even new. I think MIT had the McGovern Neurotech program, and we started using the word neurotech, but pretty much no one else in the world was. And there was neuroscience and there was engineering, and they were considered to be different things. But of course, there are people working in between. So anyway, it's great to see that it's sort of gathered momentum in the meantime. So yeah, I'm going to be telling you about, well, yeah, okay, watching thought. That's sort of the catchy title, if you like. I guess we can um, discuss whether what we're actually watching thought. And we use light to look at the nervous system. So I'll tell you a bit about that. When we look at the nervous system, we normally need to modify it in some way to make it visible to us, and we use tools that are generally sort of provided by, we use genetic tools which are provided by viruses to do that. So I'm going to tell you about some of our sort of recent attempts to use those techniques to probe brain function, and also to look at in the mouse basically sort of how brain function goes wrong. And in particular, we're going to look at a mouse model of Alzheimer's disease. Okay, so watching thought. All right. Okay, so I would sort of refer to this general area as optical neurotechnology. And I just wanted to say a few words about sort of optical neurotechnology and the prospects for optical neurotechnology at the outset. So several types of optical neurotechnology. Sort of you can use light to stimulate the brain, okay? And the main way that we think of doing that is something that's called optogenetics. So basically, we can see a sort of little example here. This was in its modern incarnation, developed by Ed Boyden and Karol Gieseroth in around sort of 2004-5. But there were a few attempts, by others in the sort of decade or so sort of preceding that. So the idea here is that you modify the function of a neuron. Again, you normally using a virus, or you can also do sort of transgenic mouse breeding to sort of breed mutant mice, which have got basically sort of artificially induced sort of proteins in, and these come from sort of various organisms, sort of deep sea organisms, etc., where they've been sort of found and sort of extracted and reproduced. One of them is channel rhodopsin, and that's a way to activate neurons. So basically, you can insert into the membranes of cells this channel rhodopsin protein, which is a light-activated cation channel, and basically allows these ions here, sort of potassium, sodium, etc., to pass through. You turn the light on, particular wavelength, 470 nanometers, and the ions will pass through, and neurons membranes will become depolarized, and they can fire action potentials. So you can cause neurons to sort of to fire action potentials or spikes. There are also inhibitory ones as well, and NPHR, and that's actually the full name is halorhodopsin, is basically a light-activated chlorine pump, and that has the opposite effect. You can basically use it to inhibit a neuron. And then there's ARCH, which is a light-activated proton pump. So you can see the proton, we've got chloride going in one direction, but that's a negative. So hydrogen going in the other direction, it's a positive, has the same effect. So it's basically inhibiting the neuron. So we've got ways to excite neurons and to inhibit them using light. And I'm not really, actually, hang on, is something going wrong here? No, okay, that's fine. Sorry. So basically, we have ways to stimulate neurons using light. And I'll come back to this right at the very end, in fact. The other way we can use light, okay, is to measure brain function, to probe or to read or measure brain function. And there's a few technologies here. There are technologies, so the optogenetics so far has been mostly used in mice. There's an exception. There are some ongoing trials in the retina. Okay, so you can basically have to inject basically gene vectors into the eye of a patient, and that is actually there are actually ongoing trials on that. So in principle, is actually translatable to humans, but most of us wouldn't want to have viruses injected in our brains, right? So it's not near term. Let's put it that way. FNIRS, functional near infrared spectroscopy, is basically a way of using light to read out infrared light to read out brain function. It's sort of like a bit like functional magnetic resonance imaging. It's sort of an optical version of that, but you can have a little helmet like that and sort of walk around. So it's got advantages over fMRI, not quite as detailed spatial resolution. And then there's the more detailed sort of version where, and I'm showing the version that we're not using here, just for contrast, where we can use fluorescence imaging. And I'll tell you a bit more about fluorescence in a minute. But here, you can see this little microscope that someone's built, which is actually attached to the head of a mouse. It weighs around 2 grams or so. So a mouse can sort of carry about 10% of its body weight on its head. So a mouse weighs about 20 grams or so. So you can sort of carry about 2 grams with a little backpack there. This is called the miniscope, and it's basically these little system, little camera in there, and then it's just sort of sending effectively a wire up, and it's got an LED to sort of stimulate, and basically the photons go into the brain. Fluorescence basically causes, basically have an electron which is sort of absorbing a photon, going up in energy level, it drops back down again, it gives off a photon that we can then detect with this little CMOS camera chip. And then you can see sort of slightly blurry images. But the neat thing about it is you can see blurry images of the brain in an animal which is actually moving around. Now, we'll see some examples in a minute of things which are much less blurry images. So I think we have in some ways a better technology, although ours is not so readily amenable to be put onto the head of a mouse that can move around. So we have other ways to do that. So that's sort of the techniques you might use there. Now, at this point, I sort of wanted to talk about, when might we use and why might we use these techniques? And so really, I say what we're doing is precision circuit neuroscience. So if we want to solve a problem, and many of the things we're looking at are what we call sort of neural circuit disorders, whether they are things like Alzheimer's disease, which are sort of, widespread degradation of neural circuits, or whether they're neurodevelopmental disorders, which may have very specific changes in neural circuits. These are all things which affect neural circuits, and the neural circuit aspect, I guess, is the real sort of functional consequence of it. So my sort of ethos in this is to make targeted interventions in small animal models of neural circuit disorders using the best technologies that we have at hand. So for changing neural circuits, that's optogenetics. So we can basically target, in fact, I didn't tell you how, but we can target individual neurons with optogenetics, or genetically defined cell types, etc. So we have a lot of control using that. And then for readout, we can either use the 1 photon calcium imaging I just told you about before. We can have 2 photon calcium imaging, calcium proxy for neural activity. Whenever a neuron fires an action potential, there's an influx of calcium, so it's a good way to sort of read out activity. We can use electrophysiological methods. So there's in vivo multi-electrode array recording techniques, something called neuropixels, which is this sort of 1,000 electrode array you can stick into the brain, or patch clamp electrophysiology, which is instead recording from one cell, but using a little micropipette sort of sucked onto the side of the cell, so you can get sort of sub-threshold signals and really, in fact, even sort of measure pharmacological aspects of the cell at that sort of resolution. So these are the sort of the gold standard measurement techniques, but you can't use them in humans. So the idea is to sort of develop your therapeutic intervention using this approach and then translate it to human by replacing these interventions. So for instance, you can replace optogenetics by other stimulation approaches. It might be deep brain electrical stimulation, transcranial electrical or magnetic stimulation. An example of that is temporal interference stimulation, which was sort of developed by Neil Grossman, or focused ultrasound stimulation. And that's something that we're actually we're very interested in, and that I'm working on in collaboration with a collaborator up in Nottingham, Jeong Jung. And then in terms of readout, basically we have the things which are applicable to humans. We've got fMRI, EEG, FNIRS we just talked about. The last one I think is particularly interesting, MEG, magnetoencephalography. Because the trouble with fMRI, very slow temporal resolution, you know, it's not going to get it, the fast things sort of, really involved in many aspects of thought. EEG does, but it's, really sort of surface only, and sort of very, very limited in sort of spatial resolution. FNIR is limited both spatially and temporally. MEG, you can image, all throughout the depth of the brain. It's not got single neuron resolution, but if we want to get at particular deep brain signals, and I've got an example I'm very interested in, sharp wave ripples in the hippocampus. That's a technology that we can use to get at that. So that may be a method that we can actually use for some of the readout things as well. So that's just sort of a general sort of overview of the approach. So the idea is you do the work in the small animal model. You make sure that you can do exactly what you want to do in the most precise way. That's the most precise way to test it. And then you sort of work on mapping it over to sort of human for translation. Okay, now sort of stepping back a bit from that, very for a high level now. You probably all know this book, Through the Looking Glass, Alice in Wonderland by Lewis Carroll. So through what Alice saw through her looking glass, her mirror in her case, was a wonderful world of talking rabbits and cards that were playing croquet and things like this. We also look through a looking glass in a sense, and we also see a sort of a weird and wonderful world of neural circuits of the type that you can see there. I'm going to show that slide a few times, and so I'm going to explain to you exactly what's in that. But other neural circuits we're looking at. Now, actually, I live just south of Guildford, about an hour sort of south of London. And so Lewis Carroll wrote these stories in Oxford. Actually, I did my PhD in Oxford, and I was in Corpus Christi College, and we could look over the back to the garden where in Christchurch College, where actually those stories were told. But then I went running last year, and I found this statue in Guildford, which is actually, it turns out that's Lewis Carroll's. He lived, when he wasn't in Oxford, he lived in Guildford, and that's his house, and the remains of the garden basically. And there was this statue erected there of Alice going through the looking glass. So I thought it was kind of nice. So I sort of like to show it. So that's her looking glass was actually a mirror, but of course you go through it. So our looking glass looks very different. Okay, this is what our looking glass looks like. It is a massive piece of glass. Okay, this is a 2-photon mesoscope. Okay, I say mesoscope rather than microscope. A 2-photon microscope. I'll show you some of the details in a second, but basically you can sort of image around 1 millimeter field of view. With this, we can image a 5 millimeter field of view, which is a big chunk of the mouse cortex, surface of the mouse. The mouse brain is about 11 by 13 millimeters, sort of, if you look at it. So we're going to get a big chunk of it that way. So the back end of this, we'll see it is basically a microscope. This thing here is about the same size as your head. Okay, it cost $200,000 to have it made. It was actually quite annoying because when we got the big grant to do this just after the pandemic, and the pound was plunging, and inflation was going skyrocketing, and there was an international supply run on various glasses and things apparently. So the company agreed to make this for us. Basically turned around and said, "Oh no, it cost you another $100,000." But of course, the grant funding doesn't. You can't go back to the grant agency and say we need another 100,000. So we ended up managing to get 4 other groups, sort of Carl Deisler's group and a few others, to make a purchase at the same time, actually. And so they gave it to us for the original cost, and then we put it together, and we somehow managed to build this microscope and get it going. So I'll show you in a few things about what this microscope can do over the next 20 minutes or so. This is a microscope for doing multiphoton microscopy. Okay, so I'll start by telling you what single photon microscopy is. So single photon fluorescence. Okay, we've got an atom of some kind in a fluorophore. There's an electron, absorbs a photon, might be a blue wavelength photon. It goes up this energy level. It then relaxes. It then comes back down very shortly afterwards, and gives off a photon, which we then see. So that's what we see through our. Now in our microscopes, it's actually normally a photomultiplier, so with very high quantum efficiency sort of photomultiplier tube, but it could be a camera, or it could even be just sort of naked eye. So that's what we see there. And sort of note that the wavelength here has got to be just slightly longer. So basically, it's just slightly lower energy photon. Just you lose a little bit of energy sort of in that process. And as to that sort of blurry image we saw before. Now, two photon fluorescence is a bit like that. I'm telling you this for a reason, so we will need it as a tool, right? So two photon fluorescence involves the simultaneous absorption of a pair of photons. Okay. Now, so each of those photons have got half the energy, okay, and therefore they double the wavelength. Now, longer wavelength light penetrates deeper through tissue. So we can go much deeper through tissue with this approach, about 500 microns, 600 microns deep into tissue in practice. And then it's still this single photon gets given off, which we then observe. Now, the physics of this process are a little different. So basically, this is sort of illumination of a little vial of fluorescent dye from the side. This is the single photon version above, and you can see, looking from the side, you can see light from all throughout. So it's focused onto this plane in the middle there, but we can see light well away from the focal plane, right? So single photon fluorescence is not really confined to sort of one location. In fact, it might be 80 or 100 microns. We might have a big sort of spread outside there. That little point corresponds to where the light coming from for the two photon fluorescence. So two photon fluorescence is confined to this very narrow volume around the focal point, around the volume of the wavelength cubed. Okay, so it's for the optical wavelengths we look at, it's about 0.4 microns laterally and a couple of microns in depth. So we've got this little point, and we can move that point around. So we can basically scan this point around in 3D in the brain to build up images. The nice thing about it being so small is if we observe a photon, we know where our point was. And for instance, if it was in a neuron, we know that photon came from that neuron and from nowhere else. It basically becomes a really precise way to read things out. And then if we do that, we can sort of scan that around. We can see things like this. So this is an image that was taken in the lab about 14 years ago now, actually. So basically, there is a layer 5 pyramidal cells in a mouse expressing green fluorescent protein. And actually, we image basically frames from the top, and we lower the focal distance. We actually physically lower the objective lens with a little stepper motor, and then I put all that together. So we've taken that sort of 3D stack, and I've put it together. I've made a rotating maximum intensity projection. So what you're seeing there. So that's the kind of thing we can see with 2 photon microscopy. It doesn't stop there. We can go on to 3 photon microscopy, and I'm not going to show you that. There's another talk I can give on 3 photon, but we have a 3 photon microscope in the department. We can see around 1.2 millimeters deep into the brain with that. Okay, so now sort of on with the talk itself. So basically, Alzheimer's disease is the sort of circuit condition, if you like, we're interested in treating there. Why? It's the leading cause of death in the UK now since 2015, excluding a brief period of where COVID-19 took over, but it's back. Alzheimer's is now back. And that's actually remarkable, because if you think about Alzheimer's, you don't think about people dying of it so much. I mean, dying with it, or but long periods of time where they're actually they're suffering, etc. So the fact that it's actually the leading cause of death means it's a big thing, because it has much greater impact beyond that. So it's got greater economic impact than cancer and heart disease combined. Quarter of all hospital beds are occupied by people living with dementia, and much of which is Alzheimer's. So it's a major and growing problem for society. So we need to deal with it, and we don't have any drugs for it, not useful ones at the moment. So what happens to brain circuitry during the progression of Alzheimer's disease? So basically, the first areas affected are the hippocampus and some connected structures, such as the entorhinal cortex. And there are 2 things that happen in them. You have these amyloid plaques, sort of like this here, which are sort of in the extracellular sort of space. Effectively, they're sort of clumps of misfolded protein that sort of aggregate. And then also you have these tau fibrils, which are found inside neurons. So it's these 2 different aspects of the pathology. The tau fibrils will eventually kill the neuron, right? Actually, reasonably quickly once they start to occur. the amyloid plaques, well, actually, probably even the soluble amyloid, which is forming into the plaques, is more likely actually the original problem. That directly sort of affecting neurons, and you can see like synapses get destroyed effectively, and sort of neurons damaged. So basically, there is this whole complicated sort of chemical cascade of events. It's not really clear. There's a chicken and egg situation. It's not so much clear if amyloid leads to tau or tau leads to amyloid. It's probably amyloid is a bit before tau in the process, but there's definitely sort of interlinked. Um, Ultimately, they lead to synaptic dysfunction. There are circuit adaptations in there as well. So basically, the brain has all these sort of homeostatic sort of adaptation mechanisms in it, tissue adaptation mechanisms. So they're sort of operating at the same time. And then ultimately, you get impaired memory and cognition. And really, that's what we want. So the sort of simplified view of it is we've got amyloid beta. It causes aberrant excitatory neuronal activity. There is compensatory inhibitory mechanisms. But that whole thing together means you've got this sort of disrupted network activity. And so what we were interested in saying is, well, actually, what's happening? What is happening in that network? How does it lead to changes in behavior? So to do that, we had to develop this platform which allowed us to do mouse behavior, and imaging at the same time. And so this is the system. Now, one of the catches is we can't image as deep as the hippocampus with 2-photon imaging. Hippocampus is about 2 millimeters deep, and we can image down to say 600 microns or so. So what we do instead is we aspirate a little bit of overlying cortex. It's extrastriate visual, sort of secondary visual cortex. Okay, so hopefully, I don't know, maybe not central visual in the mouse. I don't know. And we do that down to the corpus callosum, and we put a little cover glass at the bottom, and this is done as a sort of chronic surgery, so the animals are then recover. We do a viral injection at the same time, and in this case, we've injected a green calcium dye, sort of calcium indicator rather, GCAMP6 in the latter stuff. We've got successively later generations of this technology, and then a red one, mRuby. mRuby here is basically just marking the structure, and that's useful for doing movement correction and things like that. In some of the later things I'll tell you about, we're using that not just to mark the structure, but actually also to label particular cell classes as well. So we can have it as a sort of multi-channel thing. And this is kind of what we see. We see these sort of cells flashing away in green there as as they fire action potentials, as they talk to each other. Now, one nice thing we can do here as well is we inject another dye into the peritoneal cavity of the mouse that then basically goes into the brain, lasts for about a week. And at another wavelength, down at 720 nanometers, we can actually use that to label the amyloid plaque. So basically, the first few days we do these experiments, we map the brain of the mouse. We sort of map the area where the virus has sort of caused expression of these genetically encodable calcium indicator, the GCAM6. And the other areas, and we can see for every neuron we're imaging, we can then see where are the amyloid plaques around it. And for instance, if we see some particular disruption, say a particular neuron has a impaired spatial information or something like this, we can then say, well, actually, did that relate to how close it was to plaques? And we'll see. I think I've deleted those slides because it was too much, but basically we have the ability to sort of really relate these things together. And we do this in a behaving mouse. Okay, so our mouse, in this case, the mouse is actually just running around in a circle, but we have other things where we can have them do little bimazes and things. Now, what's happening here? This is our microscope. This is the sort of mouse is sort of head fixed here. It's running on this little carbon fiber maze there, which is floating on an airbed. So it's actually the mouse stays in one place and it moves its paws and it actually sort of navigates around this little maze. In this case, you can see there's just a little circular track that likes running around. We've got other configurations as well. That enables us to have the best optical access to the brain because our equipment weighs a lot more than 2 grams. It weighs about 500 kilograms. So we can't have it on the head of a mouse. But we can nevertheless have the mouse move around. And what we're able to do is image activity of populations of neurons. So here in these experiments, this is when we've got a very small field of view. It's only 500 microns in this examples. And we're able to image about 300 neurons at a time in those examples. You can see an example of one of the images over on the left here with in purple these amyloid plaques. We identify regions of interest corresponding to the individual cells. We pull out the calcium time series from each of these, and you can see that over on the right here. So each time you see these little calcium transients here, that's an action potential induced calcium influx. So that basically corresponds to one or more action potentials. Actually, it's often not one. It might be little bursts of action potentials. So what we actually do is we apply this deconvolution procedure to that detects the events and also their amplitudes. And we generate this sort of rastergram you can see there, basically like spike trains, but the spikes have amplitudes. So it might actually correspond to 1, 2, 3 action potentials, etc. And then we're doing this in a mouse running around. Now, the first thing we see is so-called place cells. So we're doing this in the hippocampus. Hippocampus is where you find place cells. So place cells were discovered by John O'Keefe, who's over at UCL, Nobel laureate for this discovery. And basically, they're cells that will respond in an animal when the animal is in a particular location in the environment. In this case, or in this circular track, you can see 5 very typical cells here. That this one responds every time. So I'm putting a little dot down when there's a calcium transient according to the location on the track where the animal was, and you can see this cell likes this location, this cell likes this one, this cell likes this one, and in fact if you unroll those laps, you can just sort of see the rasters sort of going down there, but actually they're remarkably consistent. There's a little bit of kind of like moving from side to side, but not in any particular direction. So we're able to sort of map place cells. Now, then the next question is, well, how did that differ between the Alzheimer mice and their wild type sibling control? So actually, the way we do this, the mouse breeding, so basically the Alzheimer gene. So it's actually this one is called the 5XFAD mouse. It's got 5 mutant human, basically genes corresponding to human familial early onset Alzheimer's disease. So there are 3 in the amyloid precursor protein pathway, and 2 in another pathway. And but when we're breeding these, you have basically a quarter of the population will get those, and then you've got a quarter that will actually just be the wild type controls. So we have the same together. Littermates that we can use as controls. So some will be Alzheimer mice, some will just be perfectly normal. And we can compare them. And we do this in both young and old mice. Now, this is a slightly busy slide. I probably should make a simplified version of this. But basically, we have young mice, we have old mice, we have wild type mice, we have Alzheimer mice. And we've got thousands and thousands of neurons. So we've got really good statistical sampling of what's going on. And what we're doing here is basically looking at fire. You can think of this as firing rate on this x-axis here. Looking at the distribution of firing rate. This is the cumulative plot which corresponds to these distributions on the left there. The key thing is, so now what we realized is we have to, so the mouse is running around in a circle. Sometimes it runs, sometimes it stops, okay? If we roll all of that data in together, it didn't make any sense. But if we separated out the times when it stopped and the times when it's running, it suddenly made a lot of sense. And what seems to happen is that in the Alzheimer mice, when the animal stopped, the firing rate increases. So there's an increase in sort of like the baseline activity in Alzheimer's disease, sort of hyperactivity of the neurons. So actually elevated firing rate. When the mice are running, it's a different story. There's actually a decrease in a comparative decrease in the firing rate. So the Alzheimer mice, and particularly the old ones, right, are not able to increase their firing rate in response to changes in their inputs. So normally, animals running around the track, this cell, for instance, there might be, it might go through its place field, in which case it should start firing more. The cells should fire more anyway when the mouse runs faster. In general, these cells have some kind of speed tuning as well. And those Alzheimer mice, the cells in the Alzheimer mice, and particularly the old ones, basically just can't increase their firing rate. So we think what's happening here is it's sort of dynamic range. So there's a reduction in the ability of the neurons in Alzheimer's to have sort of dynamic range, to be able to sort of respond to changes in their inputs. So the sort of probably in compensation for losing synapses and things. So there's these compensation mechanisms in the tissue that they're actually pushing the baseline firing up to maybe keep the average firing the same, but the firing rate that they can go to is actually decreased. And correspondingly, we can use very quantitative tools like mutual information. So this comes from information theory, so a good engineering tool. We can measure the information about spatial location using Shannon's formula. I won't go into the details of how. And you can see basically this is a cumulative plot. So cumulative plots like lower is on the left there, right? And basically in the AD mice, much lower mutual information is the key thing. So basically, the cells are not as good at coding information about spatial location, which is what this area should be about spatial memory, right? So yeah, so we have so this has given us some sort of good insights into what's going on in these mice. Okay, so next phase of the talk. So that was all done with this microscope, slightly old-fashioned microscope now. We were very excited when we got it. 500 micron field of view. We can image a few hundred cells. Then we started thinking about what could we do if we could image much bigger areas of the mouse brain at the same time. And we started building this microscope. So I've explained to you already about the objective lens, sort of 5mm field of view, had a very sort of special customized optical design. It's also got two scan paths. So basically we can take two fields of view, we can put them next to each other to have the bigger area, or we can separate them and sort of look at different areas of the brain at the same time. Yeah, so this is just a few more pictures of what this microscope looks like, sort of slightly busy optical table. And again, we can use this with behaving mouse. Again, in this case, this is the same task, basically a mouse running around in a circle. Where one key thing here, so we actually, just a nice little touch that my postdoc introduced. We're monitoring the eye position of the animal, which is actually very useful. We can monitor the diameter of the pupil. This is actually being done through the walls of the cage, okay? So we can do it at infrared wavelength and get a good enough image to actually just see it through the walls of that carbon fiber cage. Yeah, so, okay. Now, then the other thing is we need to genetically modify those cells, okay, in order to be able to see them. The strategy we were using before involves directly injecting a small volume of virus into the brain of the animal, and it labels about sort of 400 micron sort of diameter volume. And that's going to be no good if we want to image a 5mm field of view, right? We'd have to do many injections, and they'd all be a bit different, and it wouldn't be very good. So we've been working on a few alternative approaches. Now, the other thing we wanted to do is we wanted to label excitatory neurons and inhibitory neurons separately to be able to monitor two channels. And one thing we had to, there's a sort of little subtlety there. You want to avoid competition between. So actually, the viruses can compete with sort of labeling different neurons, and that can lead to sort of inhomogeneous sort of labeling. So what we've done here is we label excitatory neurons with GCAM8S. It's the best calcium indicator we've got. Basically, very big signal, slightly slow. And then inhibitory neurons we label with GCAM8M. It's a slightly faster version of the same thing, not quite as big a signal, but it's still pretty good. And then also together with that, this MSI-RFP2, which is basically a red fluorescent protein. And so the idea is, so we've got independently labeled the excitatory and the inhibitory pathways. The inhibitory ones have got red and green. The excitatory ones have just got green. So we can tell them apart based upon the red channel in our microscope images. That's what this image was from. It was one of our attempts to do that. The other thing we did with that, this is actually a little bit advanced further again. There are new types of viruses which cross the blood-brain barrier. Okay, so rather than injecting into the brain, theory, we can actually get away with just doing an intravenous injection and it labels the entire brain. And that's what was done in this image there. But that one was actually with GFP, green fluorescent protein, which is really, really strong. Um, we have had that work. We have it working with GCamp, but basically there is a loss as you go across the blood-brain barrier. So it's not quite as good as injecting it directly into the brain. But the GCam, so the GFP looks absolutely gorgeous. So you see the sort of the resolution we can get with images there. And we're able to do that to show basically that we can get these two channels. So you can see here's a green channel and our red channel, and we can see that we've got a bunch of excitatory neurons, inhibitory neurons. We can sort of monitor the two populations separately. But then how do we get it in? So we tried a few different ways. We tried ventricular injection, so you can inject it into the ventricles. Turns out that's great for labeling the hippocampus, but actually we wanted to monitor the cortex here, so it wasn't so good for that. We can inject it across the blood-brain barrier. We inject it into the tail vein, and then it crosses the blood-brain barrier. So that, as I said, we have some efficiency loss. Also, the problem we've got really here is it's labeling layer 5 cells quite well, but they're actually pretty deep. They're at the limits of the depth that we can actually image. We actually want to look at the layer 2-3 cells, which are around sort of 400 microns or so deep in the cortex, and they're actually not so well labeled. So we're working this. This technique was developed by Viviana Gradinaru and Caltech, and we're sort of collaborating with them to try and optimize that further. But in the meantime, we actually found a solution that works well, which is basically just going back to injecting it directly into the brain, but very, very slowly, of a very large volume. And it turns out it works. No one else seems to have noticed this, but actually we can label the entire brain just by injecting into the cortex very, very slowly. And it's pretty well. So that's what we're using at the moment. And those are images. That's actually an in vivo image taken from the mesoscope. In fact, I'll show you a few more examples of those in a minute. Should be. Yeah. Okay. And then, all right. So that's 5mm field of view. In fact, I've jumped on to the next thing. So, we're not satisfied with 5mm field of view. So we've got a collaboration with a group at the Suzhou Institute of Biomedical Engineering in China, in Suzhou. It's Professor Yang Gong, who are basically very good at designing lenses and fabricating lenses. They can sort of make the lenses. So we're hoping to avoid the difficulty we have with the American lens company this time. And they've made this 8mm field of view lens. So we've been over there doing experiments with it. So that's their lens. It's actually a little bit smaller, but there's still some improvements we can make to it. Then this is kind of the data we get out of that. So this is basically a whole mouse brain, right? This is green fluorescent protein. So you can see basically we can take regions across the entire mouse brain and image them, and we get this sort of beautiful localization. These are the individual neurons, how many individual neurons we can pull out. And we can do that both at sort of very coarse resolution to image many neurons, but also at very fine resolution to image dendritic spines. We can image vasculature. So here's an example on the bottom left here with vasculature across the entire brain. This one here is basically a 7x2mm sort of field of strip of that we're imaging activity in. Now you see these. It sort of looks a little bit like it's sort of out of focus fluorescence. It's actually just that it's a very big area, and we kind of zoomed out quite a bit. If you go into that and look at a small area of that, you can see this one on the right here. Then we can actually localize individual neurons, and we were able to localize 7,600 neurons from the brain in that example. You can see there's one in the middle here. This is sort of another example, basically, where we actually see the cells a bit more clearly. And then in that same system, we can go right down to the level of spines. Okay, so to image neurons, you don't really need, neurons diameter might be sort of 15 microns or something like that. So you don't really need to be down at the sort of the micron sort of scale to image activity in the neuron. But if you want to look at spines and these sort of synaptic effects, then you really do need to, and we're able to still get this very high spatial resolution at the same time. And well, at the same as I said, imaging 7,600 neurons, and you can see sort of an example here of the kind of the signal to noise ratio of the traces we can sort of get from them. So basically, we are now able to image functionally from 7,600, but that was one of the first experiments we've done. So I think we'll be able to get 10,000 pretty quickly, to be honest. We have plans to get to a factor of 10 beyond that. Okay, by instead of using normal 2-photon microscopy, we can do something called Bessel imaging. Okay, so with an ordinary 2-photon microscope, you get a very small point spread function. You're scanning a point around. Okay, and that limits your speed. So we can scan our point around, and we might be able to do about 7 or 8 hertz across a big plane. But if we then tried to then move that down and do like volumes to go down in depth, that would be divided by, it would basically go down much more quickly. And I think 7 hertz or so is I think the minimum we need to do sort of good calcium imaging. The idea with Bessel imaging is to turn your point into a pencil, basically, and then scan that pencil around. Okay, and then the idea is to you've got to do a bit of computation then. So you've got to do demixing the signals, a bit like you might have to with that single photon imaging I showed you before. So you don't know where in depth the signals came from. They might be from anywhere within, say, 100 microns, but you can demix those using non-negative matrix factorization, independent component analysis, those kinds of algorithms. And so that just add a bit of computational stuff in there. And so, using a combination of these, we should be able to get, my aim is 100,000 neurons. I guess we should be able to get 100,000 neurons with this kind of approach. And then finally, just to sort of bring it back to the behavioral task. So far, I showed you things with a mouse just running around in a circle, but wouldn't it be great if we could actually ask the mouse whether it knew what it was doing? And we have a task to do that. So we basically have a modified version of this where the mouse runs down a central corridor, and it turns left. So if it turns left one time, then the next time it comes around, it's got to turn right. So it gets rewards, and in order to get its rewards, it basically has to alternate correctly. If it doesn't know how to do the task, it will typically just go left, 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 and then it would get a maximum one reward, right? So it's actually got to sort of alternate. Now, to avoid it just learning to do a figure of 8, we have a delay period in. So we've got these little servo motors that you can see here, which basically drop these tabs down and freeze the environment. So the animal comes back around here. And when it comes back around here, this stops down until a delay period has passed. It might be 5 seconds, or we have times between 2 and 10 seconds, basically. We randomize it from trial to trial. That means the animal's got to sit there and wait until that time's passed, thinking about what it's going to do next, and then go forward and do it, right? So it kind of can't do it by sort of habitual behavior. And you can see sort of example of this with the mouse sort of being around. You see the little servo motors that should drop down, right? Pause. Yep, let's go. And of course, we can image while we're doing that, and we have. So here's an example of mesoscopic imaging, from a mouse while it's doing this task. So you can see the imaging up there. There's a little thing sort of scanning down is a sort of artifact that we're trying to get rid of. We will do that soon. This is the trajectory that's traced out behaviorally, and you can see like the high quality sort of time series we're getting from, that's from these individual neurons, these tiny little dots. We take out the time series of those, we get these kind of things sort of flashing away. So that's what we're doing in the mouse model. And where do we want to go next with this? So basically, firstly, we're wanting to take it towards looking at brain-wide mechanisms of memory consolidation. So what I'm really interested in is, when you learn something, let's say you're studying something sort of during the day, you sleep overnight, what happens is you've got this sort of memory consolidation process. And it's initially sort of dependent on the hippocampus. Once you've learned the information for long enough, in human, that's about 3 years, in a mouse, it's 21 days, then the information becomes independent of the hippocampus. But what's happening, I think, is the representation of this information in the neocortex is being reorganized continually as part of this memory consolidation process. In a way that, as we learn things, we turn our experiences into gist and ultimately facts, right? And that, there's this reorganization of the representations in the brain as that happens. How does that happen? We don't know anything about the mechanisms, really. It's something that you have to be able to look at whole brain scale to be able to look at. And that's exactly the question we're looking at. We're putting electrodes into the hippocampus to monitor these signals called sharp wave ripples that in sleep, you have this replay of experiences contained within the sharp wave ripples, these little 120, 180 hertz sort of oscillations that happen about 100 milliseconds long. We can detect them. And then we're looking at, well, what actually happens across the cortex, if we can sort of like trigger our imaging then on that and look across the cortex as that's happening. So how do these neocortical representations reorganize with consolidation? Or if you learn something new, then you think that actually the representation of the old things must actually change as well to take into account link ups with the new information. So those are kind of the basic neuroscience questions we're asking here. And then, can we use that knowledge to try and develop interventions? So Alzheimer's disease, obviously, memory disorder, can we actually have a therapy based upon improving memory? So to do that, we're actually using a closed loop optogenetics model. So we detect these sharp wave ripples, we turn on an LED, which will then basically enhance the activity of neurons just triggered to that sharp wave ripple process. So it's sort of a closed loop neuromodulation, which we can do in a mouse model. But how do we do that in a human? So ultimately, the idea is to replace the electrode in the hippocampus with MEG, magnetoencephalography. There's this new variant of it called OPM MEG, optically pumped magnetometer MEG, where you can have basically effectively a little bike helmet. You can sort of move around. Still got to be in a Faraday cage, unfortunately. And then focused ultrasound to replace the optogenetics. But there's a lot of science that needs to be done to see, well, actually, we don't really know what focused ultrasound is doing. And, we're sort of doing this in mouse. One of the catches is the frequencies you've got to use in the mouse, because the mouse skull is a different size to the human skull, are different. And so there's a lot of sort of science that needs to be done there. Okay. And then finally, there's a bunch of people in my group who sort of help with this work. They're all up there. We've had funding from a few sources. You see BBSRC, Wellcome Trust, EPSRC, and Urine and the Michael Urine Foundation, the Urine building. We had a philanthropic donation from Michael Urine's ex-wife, in fact. And the Chen Zuckerberg Initiative, they're giving us funding as well. So yeah, thanks for listening. I went way too long, sorry.
Don't worry. Thank you so much, Professor Schultz, for presenting. Now it's our Q&A session, so please feel free to raise your hand if you have any questions.
Yeah. To induce them. Okay, firstly, we haven't yet done focused ultrasound in mice. We're starting up something with Sophie Morse, who is doing it already, although she's not doing it for that purpose. So we're hoping to do that. It's not so much that we want to induce sharp wave ripples. What we want to do is to detect the presence of a sharp wave ripple using a local field potential electrode, and then detect that online, and then switch on our stimulation technology, initially optogenetics, and then we're looking at actually doing that even in mice with focused ultrasound. And the idea is basically to enhance the excitability of those cells, so that if you do it at just the right time, the cells that are basically, when you've got the sharp wave ripple, those cells will fire and will sort of boost the effect. So the idea is to be taking advantage of the brain's own sort of memory consolidation pathways and amplifying them. Right? So we use it as an amplifier, effectively. Well, we don't know whether that will work, but it's worth a go. There's some evidence from a group of Lauren Frank in the US that it might. Yeah. So not really, because there's too many underlying bits, a kind of technology developed by others. So um, the mouse behavior platform, there's a Finnish company that did some of the key development in that, and we collaborated with them, but it's their thing. Um, the the microscope the mesoscope that we've built, the optics of that was done by someone called Spencer Smith at University of California, Santa Barbara. Um, the new one, our Chinese colleagues are sort of commercializing that system, from their optics side. But our role has been basically sort of putting it all together and using it to do something new. So in a sense, there is a kind of potential thing, which is maybe a platform for pharma. I've pitched this to pharma companies quite a few times. They show interest, but by the time they come back to you, you know, their division got axed and something else has replaced it. It's like it's an unstable business. So yeah, it's yeah, maybe we'll see. Yeah.
Hey,
Yeah.
sorry. Um, Hello.
Well, yeah. So um, that would be a different kind of treatment, wouldn't it? That would be really treating the issue. Yeah. So we would expect that an Alzheimer therapy, like drug therapy, that really worked, should do that. That's what we would predict. That's not a way they're looking at it, but that's what we would predict. Yeah. So far there are drugs that work in mouse models, right? There's quite a few. The issue is that they didn't translate to humans. But in fact, we had just prior to the pandemic, we had an agreement with Novartis to test a drug that they'd developed. Um, and then pandemic happened, and we had the drug all ground up in feed. And then the pandemic happened, and the feed sat on the floor and expired. Um, and their drug got cancelled. um, in theory, we could do that. I mean, the fact that the drug got cancelled is sort of probably doesn't matter. We can still test it. Um, there are a few drugs like that that actually sort of do restore function in mice, and so that this would be a tool that could give us more information about how that's happening, which might bias a pharma company's approach, give another piece of evidence for them to continuing development of it, for instance, and going to human testing. But ultimately, of course, you need to test it in humans. Yeah. 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 Okay. Good question. What made me go down the dementia route? I guess it was in part. Okay, so I maybe sort of step back a bit. During my PhD, I started working on the hippocampus and memory. Um, I came from engineering background before that, so I did physics, math, and electrical engineering undergraduate. Um, Went to do a computational neuroscience PhD. Um, I was working on sort of hippocampus and memory. During that time, I kind of felt that it was a bit too many synapses from the periphery, too hard to control. And so I ended up then working on vision for a while because I thought, oh, we can really control the stimuli. I can get at it scientifically. And then after a while, I came back and said, well, actually, what really got me into this field is trying to understand, cognition and memory, right? Like the sort of the big questions. And in the meantime, there'd been a lot of other things done. Grid cells come out and these kind of things. And I thought, actually, we can get at the hippocampus in a much more controllable way. So I came back to it for that reason. So we started looking at some the basic memory hippocampus work. And then basically this philanthropic donation from Mrs. Urine came through, and that was specifically targeted at was targeted at Alzheimer's, but then it was aimed at, with the Michael Urine donation, was aimed at sort of engineering and medicine, right? So hence it came towards us, and we thought, yeah, well, actually that's a great opportunity to sort of to start looking at the neurodegenerative disorder side of things. So that's how that came through. Yeah.
Hello, can you hear me? Hey. No.
Maybe memory retrieval, like which obviously is not feasible now.
What do you mean by memory retrieval? You mean, yeah.
So how the tangling of the tau protein.
Yeah. Yeah.
Amyloid,
Yeah. Yeah.
amyloid beta.
Yeah. 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 Yeah.
These cause the neuron to die.
Yeah. Yeah.
So once neurons die, information is lost.
Well, yeah, that's not quite so clear. So basically, I mean, obviously, if enough of the neurons die, that is going to be true, right? But is there a period in between where you can't recall the information, but it is there? And I think the answer is probably yes. If you can actually boost the function enough, then I think in principle, recall of memories, which might not otherwise be possible, could happen. Yeah. Yeah. Yeah.
Hello. Can you hear me?
There isn't a good model of, it's difficult because of course the thing with mice is they don't live long and you can breed many cycles, etc. That's not true with non-human primates. There is some work ongoing on a non-human primate model of Alzheimer's from Daniel Beckman at Stanford. I've seen some beautiful pictures from it. I'm not quite sure what the status of it is. There are ways to do it, but they're kind of the different model. Like for instance, you can take an animal and you can inject human amyloid proteins into the brain and see what their effects are. So that's a model, yes, to some extent, a model of amyloidosis, maybe not Alzheimer's. So you can study some aspects of it. So there's been little work done in non-human primates on this question though, so far anyway. Yeah. Yep. We mostly do this in adult mice, or maybe after about 3 weeks of age, something like this. And one of the main reasons we're using viruses is if we want to do this in the Alzheimer mouse model, doing that by mouse breeding becomes incredibly time consuming and expensive because you've got to have this sort of intersectional breeding strategy where you've got all of these crosses, and at least at London cage rates, etc., it becomes hugely expensive. It's the kind of thing that if you're from the Allen Institute or somewhere you can do, but maybe not here. We have done, there's another alternative, which is in utero electroporation, which we basically take take the embryos out, do this, basically effectively you bait them in dye and put a magnetic field across, and it basically causes um, the plasmids. In fact, you put plasmids there, and the plasmids would go into the cells, and that would start to express. And that's a strategy that we have used. It's very technically difficult, so basically we've sort of stopped doing it. But
Great. If that's
Yeah.
all,
Okay.
then thank you so much once again for. Let me just check if there's any questions in the chat. No, there's not any.
Okay.
Well, thank you so much, everyone, and thank you so much to
Okay.
Professor Schultz
Well,
for
thank you.
spending his time here speaking to everyone. Let's give him